All right, great. I'm going to make this a full screen. Okay, yeah. So it's really nice to be here again. I hope this talk is relevant to the community. This is based on some exciting work that we're doing, and also we are working on several extensions. So I'm going to focus on something we worked on over the past year, which is Bayesian product mixture models for multi-scale clustering in vector autoregressions. So sorry for the long title. I know Bayesians like shorter titles. We're hoping to come up with a more fancy title in the future. So this is joint work with Josh Lukemeier, who is my former PhD student and now a research faculty at Emory University. I'm going to focus on multivariate time series data, but the ideas in this talk is equally applicable to functional data and a lot of other different types of data sets. And so multivariate time series data obviously arise routinely in different applications, in finance, in, wearable devices, and also brain imaging. And so to model these kind of data sets, there's a rich literature, obviously, to do this. And one of the very highly used tools to do this is vector autoregressive models. So we're going to focus on vector autoregressive models for this talk, but again, the ideas are fairly generalizable to other settings. So our focus really is on heterogeneous multi-subject vector autoregressive models here. So usually, the vector autoregressive models typically focus on a single subject, and then there are some papers that focus on multi-subject vector autoregressive models. However, those papers may not necessarily accommodate the heterogeneity between different subjects, right? So our goal is to really model multivariate, multi-subject time series data, again, motivated by brain imaging applications, right? So you have brain imaging data on multiple subjects, and we would like to fit some kind of vector autoregressive models for everyone in the cohort, recognizing that there are inherent heterogeneity between subjects. Obviously, one could just go ahead and fit the same VAR model for everybody in the cohort, ignoring the heterogeneity. That never really plays out well in kind of these neuroimaging applications. And then the other option is to just fit the vector VAR model separately for each individual subject. That again fails to borrow information across subjects, right? So VAR models, again, were initially heavily motivated by econometrics, and then since then, Bayesian VAR models have seen a rich development. And recently they have been people are starting to adapt this to biomedical imaging data sets, right? And so, as I was mentioning, the vast majority of VAR models has focused on single subject analysis. And so now we would like to extend this literature to multi-subject analysis, accounting for heterogeneity across subjects. Of note, there's a recent paper by Ghosh from University of Florida that showed that you can actually establish posterior consistency for single subject Bayesian VAR modeling, so which was really encouraging result. Now, the other thing is most of the literature has focused on parametric VAR models. And parametric VAR models are really nice in terms of allowing for closed form posteriors and easy straightforward updates. But also there's also certain types of data where these parametric assumptions may not hold, right? So for example, if you look at econometric data sets, the nature of shocks in the econometric analysis may not always be Gaussian, right? So for these settings, flexible probability distributions may be required. And so one may have to resort to non-parametric Bayesian approaches to model these data sets, right? So not only are we facing the challenge of modeling multiple subjects time series data with heterogeneity, but we also would like to account for deviations from normality or non-parametric settings, right? So those are the kind of challenges that we are looking to address in this paper. So there actually has been some generalizations of VAR models to Bayesian non-parametric settings. and nonlinear settings, right? So this slide provides a snapshot of that. However, most of this Bayesian non-parametric VAR modeling was primarily focused for single subject data, and there are extremely limited theoretical investigations for these type of single subject non-parametric Bayesian VAR models. So our focus is to develop multi-subject non-parametric Bayesian VAR models accounting for heterogeneity, providing theoretical guarantees, and providing computational flexibility. Now, multi-subject VAR modeling is inherently a difficult problem because now instead of just fitting one VAR model for one subject or one VAR model for everybody in the population, you get to fit n different VAR models for n different subjects. Obviously, computationally, that's very challenging, right? So we need to kind of somehow pool information across subjects while accounting for heterogeneity. There has been some literature on multi-subject VAR modeling, but these typically don't do a great job in accounting for heterogeneity. They assume known class labels or known subgroups, and so that's where we would like to kind of relax these assumptions, and instead of assuming known subgroups in these cohort of population, we would like to learn these subgroups in an unsupervised manner. Right, so we would really like to use some kind of a Bayesian mixture modeling to be able to do that. With that said, what are the objectives for this talk? The first one is to design a provably flexible non-parametric Bayesian VAR framework for heterogeneous multi-subject data that seamlessly allows inference, right? And so there's quite a few terms in this sentence. So first one is non-parametric Bayesian VAR modeling. The second is heterogeneous multi-subject data. And the third one is flexibility. By flexibility, what we mean is theoretical guarantees. And so, and really the way we go about doing that is something called a novel product mixture of DPMs, right? So, this obviously with this, audience today, DPMs, obviously everyone's an expert here on DPM. So what we do is we actually develop something called a product mixture of DPMs. And this actually allows for something we call multi-scale clustering or local partition. I'm going to talk about that more in the coming slides. So this is a more flexible way to account for heterogeneity than traditional or classical DPM models. And then we establish posterior consistency properties for multivariate time series data, and we develop efficient posterior computation, and then we apply this to a high-dimensional brain imaging fMRI dataset. So before moving forward, of note, there is some literature on multi-scale clustering, which is a key idea in our paper. But this is extremely limited literature, to be honest. And so there is a paper by Dunson back in 2009 on LPP, local partition processes, and then another paper by Suarez and Ghoshal in 2016 in Bayesian analysis that is closest to our work. So essentially, the idea that they use is for functional data, they propose an independent Dirichlet process model for each basis coefficient, right? So instead of doing the classical way of DP, which is just specifying the DP on the vector of all coefficients, you get to impose an independent Dirichlet process for each coefficient. And what this really does, it allows you to cluster different aspects of your data separately, right? Instead of having identical clusters, instead of forcing subjects into one cluster and forcing them to have identical elements, you get to do multi-scale clustering. You get to cluster at a local level, higher resolution level, right? And that provides you more flexibility in terms of clustering that hopefully provides you with a better computational, a better model to fit your data better, if you will. Right. And then there was some work by Petron around a similar time frame, 2009, who proposed canonical curves and to account for heterogeneity, right? So the key idea here is when looking at functions or looking at high dimensional data, there's always going to be some irregular local features that cluster very differently compared to global features that are more regular, that are more prominent, right? And so ideally, you want to cluster these global features separately and these local features separately. And so the idea here is if you don't allow for these differential clustering between these global features and local features, chances are that under a classical DP where you force these global and local features to cluster the same way across subjects, the chances are that your local features, which may not be even relevant to your data as much as the global features, may actually start to interfere with the clustering of the global features. So the clustering of the local features can actually interfere in your model fitting and can really drive down the accuracy of these models, right? So there is a real need here to recognize that not all features in the data clustered the same, and different features can cluster differentially, and then to be able to allow us to do that via this kind of a product partition, right, or an LPP model, right? So now having said that, this idea was proposed back in around 2009 in the follow-up work by Suarez and Ghoshal, but then these methods typically focus on one-dimensional functions, right? So they're considering time points, right? And they're not, they may not be straightforward to generalize to multivariate times, to multivariate time series data. And they may not be able to generalize to high-dimensional imaging data, for example, right? So, and then the other thing is these papers really did not focus on providing posterior consistency guarantees for density estimation. That is also our focus, right? So these ideas were very appealing, but there's a lot more to be done in this regard. Okay, so this is what a product mixture for multivariate density estimation looks like. So I think most of you are familiar with this kind of a kernel mixture form of the density, right? So you assume some kind of a normal kernel, and then you impose some flexible probability distribution. For this talk, it will be, let's say, Dirichlet process on mu and sigma, right? So this is the classical setup you would use for density estimation, for multivariate density estimation. Here, X is a multivariate vector. Now, instead of this, if consider partitioning the mean parameter into smaller subsets, right? So mu M1, mu M2, and so on, and consider partitioning the variance parameter into smaller subsets. And then consider imposing independent priors on each of these partitions, right? So mu M1 has these follows P, and then sigma M2 follows P sigma, and then these are assigned a Dirichlet process, right? So essentially, instead of just putting, just saying that the whole vector mu follows the Dirichlet process, what we do is you end up splitting the mu into different partitions, and then for each partition, you end up imposing a Dirichlet process. And so what this specification does, is it results in a product mixture prior. It's particularly product of DP priors, right? And so the exact form of the prior obviously depends on the way you partition your parameter space, right? So that obviously will control the way the particular form of the prior. But what this kind of partition does, it allows you to cluster these different subsets of parameters differentially in your model. So here's a very simple example, like a visual depiction, right? So if you have these, let's say the mean parameter A1, like A, and the variance parameter sigma, if you look at the classical Dirichlet process, you would cluster the mean and the covariance parameters together, right? So let's say this is a red cluster, this is a blue cluster. So all of the subjects in the red cluster will be forced to share the mean parameter and the variance parameter, right? So they will have identical mean parameters, and then they will have identical variance parameters. And then similarly for all the subjects in the blue cluster. But then here with the product mixture, we can actually allow subjects to have in the red cluster, subjects have the same mean parameter. And then in the blue cluster, they have another mean parameter. And then the variance parameters can actually cluster separately, right? So you can actually separate out the clustering for the mean parameter and the variance parameter, right? So this is what the product mixture model does. And then you can take this a step further. You can even partition this mean parameter into subsets, and you can apply this differential clustering to these subsets, right? So you can actually take it, in a finer and finer and finer levels, right? And so what this plot shows is the clustering accuracy, and this clustering accuracy across different dimensions. You start to see that the clustering accuracy is actually higher for the product of DPM models compared to the classical DPM models when there's a lot of heterogeneity in the data. And so this is just a schematic here, but hopefully this conveys some of the idea of the models. And so now coming to the actual VAR model. So remember, we have multi-subject data. So consider the data matrix Xi. So Xi is Xi1 to Xit, where Xit represents a d-dimensional vector at the tth time point. So for the motivating applications for this talk, we're going to focus on fMRI data, which is a functional magnetic resonance imaging, which is a neuroimaging data set. And Xit, again, is a d-dimensional vector, d being the number of brain regions, right? So you observe different brain regions, activity in the different brain regions, and you observe it across different time points, right? And so Xit represents this vector, so one of the key assumptions here in this talk is that d and the number of regions, d, which is a dimension of the multivariate vector, and the number of time scans t are fixed. This is not unreasonable in neuroimaging applications, but these are the assumptions we really have to make for our theory to go through. And so this is something that we will assume throughout the talk. And then consider this VAR model, which looks like this. So I think this is a common form of the VAR model. So essentially, Xit depends on the previous time point, right? And AIK is autocovariance matrix at the kth lag, and this matrix is really big. It's a D by D matrix, right? And typically the brain imaging, D would be, if we consider ROI level data, brain region level data, D would be 100 or 250 or 360, depending on the kind of brain parcellation that we're going to use. So imagine this being a 100 by 100 matrix. And now instead of just trying to solve one VAR model, we have n different VAR models, n being the number of subjects, right? So you can see the complexity of this operation. And again, we would like a way to borrow information across these n different VAR models in a very flexible way that allows for heterogeneity. So that's the goal of this operation here. And so this is what a structure of a VAR model looks like. If you are a more picture person, hopefully this schematic helps a little bit. And so essentially you have these. At the tth time point, you have the d-dimensional vector, and these are the autocovariance matrices across the k lags. Each of these matrices is a d by d matrix. K is the number of lags, which is typically assumed to be known for the theoretical purposes. And then these are the vectors at the previous time points, right? And these obviously are the error terms. So now classical methods, if we were to go ahead and use the classical Dirichlet process and just cluster these covariance matrices, now remember these matrices are obviously very high dimensional. I mean, if D is 100, then this is 100 times 100 times K. Let's say K is 1, the lag 1 operation, lag 1 VAR model. So this is a 100 by 100 matrix that has obviously 10,000 elements, and to cluster all of these elements together is under a classical DP is a pretty daunting task. And that clustering may not even be accurate because these are really high dimensional settings, right? But classical methods, they do exactly that. They assume replicated samples having fully identical parameters. And so as I was mentioning, this really may be restrictive in high dimensions, right? And these high dimensions, samples may often be effectively clustered along only a subset of directions, right? So the idea here is that when we are faced with this high dimensional object, 100 by 100 matrix, the idea is that the clustering across subjects is only informed by a subset of these parameters. And the remaining parameters really are kind of noise parameters, and they don't really contribute as much to the clustering. So ideally, we would like to separate the clustering of these informative parameters and then these non-informative parameters, so then they do not interfere with each other. So this is where we propose a class of novel Bayesian non-parametric VAR models that specifies DP mixtures on subsets of the autocovariance matrix elements. So we're going to take this autocovariance matrix elements, and we're going to take the variance parameter sigma, and we're going to specify, and we're going to break these parameters down into smaller subsets, and then specify a Dirichlet process on each subset separately, and have achieved differential clustering. So that's, again, the key idea. And so this is what we call the product of DP mixture model, so or PDPM in abbreviation. So we actually end up developing 3 variants of the model that differ in the scope of clustering and the scope of heterogeneity. So the first one is the PDPM VAR model, then the row generalized, the RG-PDPM VAR model, and then the lag generalized RG-PDPM VAR model. So the PDPM VAR model is the simplest case where we separately cluster the autocovariance matrix and then the covariance matrix sigma, right? And then the row generalized VAR model actually imposes a separate clustering for each row of the autocovariance. So this is where we are breaking down the autocovariance matrix, which is a really high dimensional object, into smaller chunks, and then we're imposing independent DP prior on the different chunks of the autocovariance matrix. And in this particular case, by chunks, we demarcate each chunk as a row. So we impose a DP separately on each row of the autocovariance matrix. And then the lag generalized PDPM VAR imposes differential clustering across lags. And we're not going to talk about it in this presentation for the sake of time. And so one really cool thing about these methods is that you can actually impose appropriate base measures in the Dirichlet processes. And this allows the processes to induce shrinkage for the autocovariance matrix elements that facilitate feature selection. And you can also do lower dimensional representations of the covariance matrix, which allows you to scale up your method to a larger number of nodes or to larger dimensions. And so getting into a little bit of math notation here, and so we'll omit the subscript I as needed. I typically denotes the subject index. And so if I denote as script theta, these autocovariance parameters across k lags, then these autocovariance parameters follows the Dirichlet process, and then these covariance parameters follow a separate Dirichlet process, right? And then the resulting prior on the parameter space is pi star, which is given by a product of these 2 different parameters, these 2 different priors, one for the autocovariance and one for the variance. And the resulting density, using the Sethuraman stick breaking representation for the Dirichlet process, looks something like this, right? So now you have really 2 different summations going on because you have 2 different stick breaking representations. And then it's possible to show that the form of the density, takes on this kind of a representation. So this, again, if you look at this for a second, you will realize it is very you'll probably recognize the underlying similarities with the regular stick breaking representation of the DP, but there are also the differences here. And then the next generalization is the row generalized PDPM VAR model. So here we actually impose a Dirichlet process separately for each row of the autocovariance matrix. And then this is the form of the RGPDPM VAR. This is what the density looks like, right? So you end up imposing for the row D prime, you end up imposing a Dirichlet process prior, and then this is what the density looks like. So now you have all these different summation terms and for the stick breaking representations. Moving on to some of the posterior consistency properties. So we assume that these D by T matrices, so now we're actually dealing with matrices, right? X1 to Xn, this is our data. And these matrices are essentially comprised of multivariate time series data. And so we assume that these matrices are IID under some true density F naught belonging to the space of densities F. And we study the convergence of the density of the posterior. We see how well the posterior converges or concentrates around the true density F naught as the sample size grows to infinity. And we measure the convergence using, Hellinger distance or L1 distance when D and T are fixed. And then we assume certain regularity conditions that are more or less standard or routinely used in Bayesian non-parametric literature. Okay, so the posterior consistency properties of these VAR models represent non-trivial extensions of the rich theoretical literature on multivariate density estimation. So obviously, there's a very rich theoretical literature on Bayesian non-parametric theory for multivariate density estimation. What we show here, we kind of extend that literature, that theory, to a multivariate time series data sets. And so the way we resolve some of the theoretical challenges are by establishing the Kullback-Leibler properties for VAR models. Because remember, VAR models have a very different likelihood than the traditional multivariate models. So we established the Kullback-Leibler properties, and then we carefully design sieves. So we have to design these sieves really carefully. And then based on those sieves, we have to derive entropy bounds and tail probabilities and so on. So the sieves constructions becomes very important for our case. And then also we show that the theory really holds for commonly used and computationally tractable base measures of DPs, right? So you could use a Laplace or a normal base measure on the DP for the mean or the autocovariance, and you could also use low-rank structures for the residual covariance. Like, for example, you could use latent factor representation. So essentially, this kind of theory shows that your method is actually practical to implement. So the first one is obviously prior support and weak consistency. So we show that under reasonable assumptions, the posterior has leads to weak consistency, right? So sorry, the prior leads to weak consistency, right? So this is what it is. And so the next step is to establish strong consistency, and the way we do that is again by using sieves, right? So if Fn is the sieve, and then we show that this kind of a construction of the sieves. This is how you would construct the sieves, right? So this, if you look at this hard enough, you might be reminded of some related literature for multivariate density estimation, but again, there are important differences for these kind of sieve constructions, right? So essentially, you want to control the maximum and minimum eigenvalues of the covariance matrix. You want to control the condition number, which is the ratio of the maximum and minimum eigenvalues. You also want to control the norm of the autocovariance matrices, and so on, right? So all of these considerations need to come into the picture. And so under using these sieves and under certain reasonable conditions, we show strong consistency, right? Which is a desirable result. And then we show that this, in some follow-up lemmas, we show that this above results hold for normal and Laplace-based measures on the autocovariance elements. And then it also holds for low-rank structures on the residual covariance. And for the posterior computation, we actually develop a Gibbs sampling approach. And we actually end up using this kind of a low rank or a latent factor representation for the model. And remember, the theory actually allows for this latent factor representation of the covariance matrices. The reason we use this latent factor representation is to really scale up the model to high dimensions, right? And so this really, I mean, we were really getting stuck with maybe dimensions less than 100 before doing this, but once we were able to do this, we were able to scale up to, 100 or slightly higher dimensions in reasonable time. I would like to take this opportunity to also present some simulation settings, the simulation results that we generate. So we actually generate data from VAR models with lag2 with different dimensions and clustering structures. And we actually consider 4 different scenarios. The first scenario, the data was generated from the PDPM. The second scenario, the data was generated from the row generalized PDPM. The third one where the data was generated from the lag generalized PDPM. And the fourth one where it's a very heterogeneous scenario where, none of the, some of the inherent assumptions in the model are not satisfied. And so then we compared to several methods, including the single subject Bayesian VAR model by Ghosh in 2018. And then also an ad hoc method where we first we combine the single subject VAR model with a form of clustering, right? So we first cluster the data into subgroups using any of the shelf hierarchical clustering approaches. And then for each cluster, we impose the parametric VAR, or we fit the parametric VAR model, right? So this method, as I said, is ad hoc and is also highly sensitive to the initial clustering, and then it cannot accommodate uncertainty related to clusterings. And so here are some results for the 4 settings. This measures the clustering accuracy. These 2 rows denote the differential sample sizes, n equal to 100 and n equal to 200. And I believe these results pertain to 100 nodes, right, and 250 time points. So you can see here that the PDPM VAR and the RGPDPM VAR do fairly well. They have a fairly high adjusted RAND index, close to 1 for setting 1, which is a simpler setting. And then for more complex settings, the RAND index can drop. But again, the RGPDPM VAR has a fairly high RAND index across most settings. And then going beyond the clustering, just looking at the estimation performance, the relative error for the autocovariance matrix, we can see that, again, the RGPDPM VAR has a fairly low relative error, which means it can accurately estimate the parameters in the model. So this is the row generalized PDPM VAR model. Again, looking at feature selection for the autocovariance matrices, again, the PDPM VAR obviously does well in setting 1 because this setting is friendly to the PDPM VAR. That's how the data was generated. But overall, we see that the RGPDPM VAR tends to do fairly well in terms of the area under the precision recall curve. And then it also does fairly well under the area under the ROC curve for feature selection. So some comments on the simulation results. Obviously, the paper has much more richer simulation settings. Feel free to take a look. But there's some comments here. The single subject VAR analysis with and without the clustering leads to considerably poor performance as expected. And so what this really tells us is if you fail to pool information across subjects, you fit the data separately for each subject, then you don't get good results. And then also if you try to pool information across subjects, but you do it in a non-systematic way or in an ad hoc way, then that can actually end up hurting you because your clustering is off, and then your incorrect clustering can actually propagate errors to other parts of your model, right? It really can mess your estimation and inference as well. And so the other result that we found was really promising is we fit our model with the low rank representation of the covariance, which allowed us to scale up to higher number of nodes. And then we compared the estimation and inference performance of that model with an unstructured covariance setting, which obviously is not as scalable to high number of nodes. And we found that, the results comparable between the low rank covariance and the unstructured covariance. The other thing that I would note here is that most of the VAR literature actually focuses on the diagonal diagonal covariance for the residual. They don't even consider unstructured covariance. And I believe that is for computational considerations, because the diagonal residual covariance really helps you in computation, in scalability for VAR models. But we found that actually imposing some kind of a non-diagonal structure allows us to do better in the VAR model scenarios. And of course, last but not the least, there are these multi-subject VAR models that I had kind of mentioned in the beginning of the talk, but they all assume known grouping information. So they cannot really be applied to these settings where we assume the grouping information of the subgroups of the clusters are really not known to us. Next, we apply the method to the analysis of Human Connectome Project neuroimaging data. So our goal is to actually investigate differences in effective connectivity between low and high fluid intelligence groups, right? So you have a low fluid intelligence group and you have a high fluid intelligence groups. And then we want to see what are the connectivity differences in between these two groups. And if you're not familiar with effective connectivity, just side note here is that under the VAR models, you can actually very easily calculate effective connectivity because those are directly tied to the autocovariance matrix in your model, right? So we select the top 10% and the bottom 10% of subjects with respect to fluid intelligence. So these are two cohorts separated by two spectrums of fluid intelligence, and we have a total of 306 subjects. We consider the fMRI data, which is a functional MRI data. So this is a multivariate time series data, and we have about 1,200 time scans, and we have 360 regions. Now, while we were tempted to directly fit the VAR model to 360 regions, it was computationally, impossible for us to do, because remember, we are not fitting one VAR model, we're fitting, n different VAR models here jointly. And so we ended up dividing the 360 regions into smaller functional networks. So each of these functional networks had anywhere between, 30, 40, 50, 60, 70 regions, right? So we made the problem more manageable. And we used a lag1 VAR model for analysis, which was backed by exploratory analysis. So if you're not familiar with functional magnetic resonance imaging data, basically you put the patient on, the patient lies here, and then you put them in the scanner. And this is a Siemens, I believe 7 Tesla scanner. And there's a really big magnet that goes around here, and it keeps recording your brain over time. And you just lay there, right? And so what you really end up getting is 3D images of the brain across time. So you really get a 4D data, right? So you get a time series of 3D images. And so then we use the RGPDPM-VAR to fit, to fit the data. And then we also use the Bayesian framework to do inference, to really determine significant differences between the high and low fluid intelligence groups. Okay, and then we compare the performance to a single subject VAR model. Okay. And so for the proposed method, these are the kind of differences that we find. So these are the 6 networks that we've broken down the brain into: the central executive, default mode, dorsal salience, somatomotor, ventral salience, and the visual network. And these are the areas that we find differences in effective connectivity under the proposed approach. And there are some networks with more differences, some with less, like the dorsal salience, has quite a few differences. And then the default mode has probably fewer differences. And so the strongest differences are actually discovered in the dorsal salience network, along with, along the diagonal. So this is actually backed by literature that has suggested that dorsal salience and attention networks to be highly relevant to fluid intelligence. So fluid intelligence is defined by thinking on your feet, right? So you are faced with a new problem all of a sudden. How do you adapt to that? How do you respond to that, right? And it is known in literature that the dorsal salience network is highly engaged during these kind of tasks. So they are associated with fluid intelligence. And we were actually able to, if you look at this table, the regions in the brain, or the connectivities in the brain with the highest differences between the low and high fluid intelligence groups mostly belong to the dorsal salience network. So in this list of the top, about 15 or 20 are, the highest differences, most of these belong to the dorsal salience, right? And so, this kind of finding matches with the existing literature. Now, unfortunately, when we fit the single subject VAR model, collate information or collapse information across subjects with a single subject VAR model, we were able to find only one significant difference between the high and low fluid intelligence groups after FDR correction. So this we feel is biologically implausible. And we feel the reason for the single subject VAR model to not find differences between these high and low fluid intelligence groups is due to the fact that it's not able to really account for the heterogeneity within subjects, between subjects for each group, right? So if you don't account for heterogeneity, then you end up losing a lot of the information, which is what precisely what might be happening here. We also looked at clustering reliability. So we actually ran the MCMC 2 times and for this fMRI data, and we show that we end up getting fairly consistent clusters. Obviously not the same. There's obviously variability when you rerun things, but we found healthy clustering reliability here. And then we also looked at biological reproducibility. So we wanted to be sure that our results are biologically reproducible, which means that if tomorrow we get a new data set, we would like our findings to hold for that new data set, right? So we actually took 2 separate sessions, like from the HCP data. So each subject actually has 2 different fMRI sessions in HCP, actually 4. So we looked at 2 of them, and we fit the model separately to the LR1 session and then the LR2 session. And we then compared results, and we found that the autocovariance elements, the correlation between the autocovariance elements estimated between these two different sessions was actually fairly consistent under the RGP-DPM-VAR model, which shows biological reproducibility. Whereas for the single subject VAR model, the correlations were much lower, which is kind of concerning because then that tells us that these results are not really generalizable to other data sets, that there is low reproducibility. So this paper is under revision, and now we are extending the product of DPMs to clustering functions, right? So, and what we're really working on is, high dimensional images as functions, and we're using this product of DPM idea to separately cluster the global and the local features in the image. And we hope that this will lead to more interpretable analysis. So right now we are working on this paper, and we are looking forward to what kind of discoveries we have or we find. But this the code for this paper, it was written in R and RCPP, and it's available in GitHub. And this research was generously supported by NIA and NIMH awards. And a shameless advertisement, my lab is looking to recruit postdocs. So if you know of any good ones, please feel free to point them along. And then I'm happy to take any questions. Thank you.
Thank you, Suprateek. And it's time for questions. If you have a question, please raise your hand, and I will call your name. Okay, it seems no questions. I just have a small question, and then maybe I can take advantage of this. So you have a product mixture of DPMs. So so then, how do you summarize the clustering, the inference on clustering? Because you have many clustering indicators. It's so how do you do that?
Yeah, that's an excellent question. So that's actually one of the difficulties or challenges with these multi-scale clustering methods, as you precisely pointed out, because you end up getting a different set of clustering for different sets of model parameters. And so the way we are able to resolve it is using some kind of a downstream or a post hoc analysis, where we would look at the similarities between each pairs of subjects, using the fitted MCMC model, and we could then create an n by n similarity matrix, right? So, and then one could apply any kind of a hierarchical clustering approach on that similarity matrix. So the idea is that the clustering at the differential scales, the multi-scale clustering, it's hard to kind of consolidate it into one clustering, but one could potentially use those fitted MCMC, fitted estimated parameters to create a similarity matrix across subjects, like n by n matrix, and then simply do some kind of a hierarchical clustering on that. Yeah, thank you.
Thank you. Any other questions from anyone else? Okay, so then let's thank our speaker again. And we have our next webinar will be May 2nd, which is Thursday, not Wednesday, due to the Labor Day. So then I will meet you a month later. And thank you, Suprateek, again. Bye.